Biotechnological methods in diagnostics using nanomaterials. Lecture topic: Biotechnological methods in diagnostic using nanomaterials. Lecturer: Professor Natalia Kozan, Ivano-Frankivsk National Medical University. Purpose, objectives, and expected outcomes. Purpose: to provide students with an understanding of modern biotechnological diagnostic methods involving nanomaterials. Objectives: to examine classes of nanomaterials, their properties, diagnostic platforms, and examples of applications in medicine. Expected outcomes: gaining theoretical knowledge in nanobiotechnology and the ability to analyze advantages, risks, and the potential of such approaches. Modern diagnostics: needs, challenges, breakthroughs. Traditional diagnostics face limitations such as long processing times, low sensitivity, and dependence on large laboratory infrastructure. Nanotechnologies enable the development of rapid, sensitive, and portable diagnostic tests. Traditional diagnostic limitations: long processing times, low sensitivity, dependence on large laboratory infrastructure. Nanotechnology solutions: rapid testing, enhanced sensitivity, portable diagnostic system. What are nanomaterials? Definition: nanomaterials are substances with particle sizes ranging from 1 to 100 nanometers. Unique properties: high surface area to volume ratio, quantum effects at nanoscale, enhanced electrical conductivity, novel magnetic behavior, altered optical properties. These unique properties make nanomaterials particularly valuable for diagnostic applications, enabling the development of highly sensitive and specific detection methods. Advantages of nanomaterials in diagnostics. Nanomaterials provide significant advancement over conventional diagnostic methods, enabling more precise, accessible, and efficient healthcare solutions. 1. High sensitivity. Detection of extremely low concentrations of biomarkers, earlier disease detection than conventional methods. 2. High specificity. Precise molecular recognition, reduced false, positive, or negative results. 3. Capability for multiplex analysis, simultaneous detection of multiple biomarkers, comprehensive diagnostic profiles from single sample. 4. Miniaturizing of diagnostic systems, reduced reagent consumption, compact portable devices. 5. Potential for point-of-care (POC) testing, rapid results in clinical or field settings, democratized access to advanced diagnostics. Bio and nanotechnologies, interdisciplinarity, integrity of field. Modern nanodiagnostics integrates multiple scientific disciplines to create innovative diagnostic solutions. For example, biotechnology, chemistry, physics, material science, informatics. Synergistic approach. The field is defined by the synergy between fundamental science and applied technologies, where nanoscale phenomena meet biological systems to solve clinical diagnostic challenges. Practical impact. The interdisciplinarity convergence enables development of novel diagnostic platforms with improved sensitivity, specificity, and accessibility compared to traditional methods. Metallic nanoparticles. Metallic nanoparticles 1 to 100 nanometers exhibit unique optical, electromagnetic, and catalytic properties due to quantum effects and high specific surface area. They are widely used in nanobiotechnology, particularly in diagnostics. Key properties: surface plasma resonance, optical sensing, magnetic behavior, separation, purification, catalytic activity, signal amplification, high surface to volume ratio, functionalization. Examples: gold nanoparticles, silver nanoparticles, magnetite. Quantum dots. Key features: quantum dots are semiconductor nanocrystals, 2 to 10 nanometers, with unique optical properties resulting from quantum effects. Their fluorescence emission color depends on particle size, making them powerful tools for biomedical imaging and multiplex detection. Carbon nanotubes and graphene in diagnostics. Carbon nanostructures. Carbon nanostructures are among the most promising materials in diagnostics. Their exceptional properties make them key components in next-generation sensors, particularly for personalized medicine and digital diagnostics. Unique properties: electrical, exceptional conductivity, sensitivity to molecular interactions. Chemical, high surface area for functionalization, reactivity control. Mechanical, strength, flexibility, and stability for durable diagnostic devices. Diagnostic applications. These carbon-based nanomaterials enable label-free detection of biomarkers, electrochemical sensing, and field effect transistor biosensors. They form the foundation for wearable diagnostic technologies and implantable monitoring systems. Polymeric and lipid nanomaterials. Polymeric and lipid-based nanomaterials represent versatile platforms for diagnostic agent delivery, offering biocompatibility, controlled release, and targetable properties. These materials can be engineered with various compositions and surface properties to optimize their diagnostic performance. Liposomes, lipid bilayer vesicles, 50 to 200 nanometers, encapsulate hydrophilic hydrophobic diagnostic agents used for drug or probe delivery. Micelles, amphiphilic nanostructures, 10 to 100 nanometers, core shell architecture, solubilization of hydrophobic compounds. Nanogels, cross-linked polymer networks responsive to environmental stimuli, pH, temperature, controlled release and targeting capabilities. Biocompatibility and functionalization. Biocompatibility, a key requirement for the medical use of nanomaterials. It means that the material does not trigger adverse immune responses, is non-toxic to cells and tissues, does not impair cellular or tissue functions. Surface functionalization, the process of modifying nanoparticle surfaces with biomolecules, antibodies, aptamers, enzymes, or chemical groups to enhance stability and biocompatibility, provide specific targeting capabilities, enable biorecognition and signal transduction. Critical importance. Biocompatibility and surface functionalization are essential steps in designing effective and safe nanomaterials for diagnostics, ensuring not only safety, but also high selectivity and reliability of biosensor platforms. Immunoassays and nanoparticles. What are immunoassays? Diagnostic methods based on the specific interaction between antigens and antibodies, providing the foundation for detecting biomolecules in clinical samples. Nanoparticle integration. The integration of nanoparticles into this system significantly enhances sensitivity, specificity, and detection speed by amplifying the signal or improving target molecule isolation. Clinical impact. Nanoparticles have made immunoassays faster, more accurate and accessible. They form the basis of technologies that revolutionized diagnostics of infectious, oncological, and metabolic diseases in clinical practice. Nanosensors, definition and function. Nanosensors are highly sensitive devices that use nanomaterials, for example, gold nanoparticles, carbon nanotubes, graphene, to detect biomolecules, toxins, viruses, or cells. They work by converting interaction with a biological target, antigen, DNA, enzyme, etc., into a measurable physical signal. Types of nanosensors. Optical sensors change color, fluorescence, or absorption reflection spectra when binding to a target. For example, LSPR, localized surface plasmon resonance. Electrochemical sensors detect changes in potential current or impedance using nanomaterial modified electrodes. Mechanical sensors use nanostructures as resonators that respond to mass or force changes from biomolecule binding. Lab on a chip with nanostructures. Lab on a chip, LOC, refers to miniaturized analytical systems that integrate several laboratory functions, sample collection, processing, analysis, on a chip the size of a few square centimeters. These systems enable automation and acceleration of diagnostics even outside laboratory settings. Key components and features. Microchannels for fluid transport and mixing, nanofabricated structures for detection and separation, integrated sensors and microelectronics. Advantages with nanostructures: enhanced sensitivity and detection limits, reduced sample and reagent volumes, NL to PL, faster analysis through shortened diffusion distances. Applications: point-of-care infectious disease diagnostics, continuous monitoring of biomarkers, environmental and food safety testing. Microfluidic systems. Definition: microfluidic systems are devices where fluids move through channels tens to hundreds of micrometers wide. Key features: they offer precise control of mixing, reactions, and sample transport at microscale volumes. Nanomaterial integration: when nanomaterials are integrated into microfluidic platforms, detection sensitivity and specificity are significantly improved, enabling more accurate and rapid diagnostics. PCR, ELISA, and nanomaterials. Traditional molecular and immunological diagnostic methods are significantly enhanced by the integration of nanomaterials, improving sensitivity, speed, and accessibility. PCR, polymerase chain reaction. Magnetic nanoparticles extract nucleic acids from biological samples, plasma, swabs, saliva. Magnetic separation removes inhibitors, improving amplification accuracy and sensitivity. Enables rapid purification and concentration of target sequences. ELISA, enzyme-linked immunosorbent assay. Traditionally enzyme-based, for example, HRP, ALP. But addition of quantum dots or gold nanoparticles enhances fluorescent or colorimetric signals, reduces detection limits 10 to 100x, increases speed, and enables multiplex analysis. Nanoparticle modified platforms allows simple visual readout of results. Virus detection. Gold nanoparticles. Antibody-conjugated gold nanoparticles are used in lateral flow assays that provide results in 15-30 minutes without lab equipment. Effective for screening SARS-CoV-2, hepatitis B, HIV, Zika virus, and others. Example: gold nanoparticles. Lateral flow tests for COVID-19 detection allow for rapid point-out care diagnosis with minimal training required. On-chip PCR. Miniaturized PCR combining magnetic RNA DNA extraction, amplification, and fluorescence readout on a single chip. Provides high sensitivity and automated processing, reducing analysis time to 30-45 minutes. Benefits: integration of nanomaterials enables enhanced sensitivity, full deployment capabilities, and faster diagnosis compared to conventional laboratory testing. Cancer diagnostics. Early detection of cancer biomarkers is crucial for treatment success. Nanomaterial-based platforms offer unprecedented sensitivity, specificity, and multiplexing capabilities for cancer diagnostics. Key biomarkers: PSA, prostate-specific antigen, prostate cancer. HER2, human epidermal growth factor receptor 2, breast cancer. CA125, ovarian cancer. Others: CEA, AFP, CYFRA211. Nanotechnology applications. Electrochemical sensors with CNT or graphene modified with antibodies. Fluorescent nanosensors with quantum dots for multiplex detection. Magnetic nanoparticles for enrichment of tumor markers in blood samples. Neurodegenerative diseases. Target biomarkers: beta amyloid, A-beta, aggregation in Alzheimer's disease, tau protein, microtubule instability, alpha-synuclein, Parkinson's disease marker. Role of nanotechnology. Nanobiosensors detect these proteins in serum, CSF, or urine. Magnetic platforms for rapid biomarker isolation. Nanogels for controlled release of prop reagents. Outcomes: detection at preclinical stages, potential for high-risk population screening, development of non-invasive disease monitoring tools. Infectious diseases. Nanomaterial-based platforms offer rapid, sensitive detection of infectious pathogens, enabling earlier diagnosis and treatment. Their field deployability has revolutionized disease management in both clinical and remote settings. Examples: tuberculosis, TB, gold nanoparticle immunoassays for ECAT6 antigen; syphilis, lateral flow tests detecting IgM and IgG antibodies; influenza, graphene and gold-based immunosensors to identify A or B virus strains; platforms, lateral flow assays, LFA, paper-based, rapid results in 10-30 minutes; microfluidic chips with magnetic separation, concentrated sample analysis; electrochemical sensors with CNT, graphene electrodes, high sensitivity. Metabolic diseases. Nanosensors for key metabolites. Glucose, nanomaterials, for example graphene, gold, iron oxide, serve as platforms for immobilizing glucose oxidase. Electrochemical sensors enable continuous or drop-based monitoring of blood glucose levels. Cholesterol. Nanoparticles coated with enzymes, cholesterol oxidase, react with cholesterols to generate a signal, electrochemical or optical. These sensors enable non-invasive monitoring of lipid profiles. Uric acid. CNT graphene-based sensors use uricase as a biorecognition element for specific detection. This allows for early detection of hyperuricemia and prevention of gout. CRISPR and nanomaterials. Enhanced delivery of CRISPR systems. Nanoparticles act as carriers for Cas9 or cgRNA complexes. Lipid nanoparticles enable efficient delivery into target cells. Gold and polymeric nanoparticles provide high stability and controlled release. Magnetic carriers allow navigation using external magnetic fields. CRISPR Diagnostics. Platforms such as Sherlock and Detector utilize Cas12 or Cas13 enzymes for highly specific recognition of DNA or RNA from pathogens. Nanomaterials amplify signal output and lower the limit of detection to the attomolar level. Multiplex diagnostics. Multiplex diagnostics enable simultaneous detection of multiple biomarkers within a single biological sample, significantly saving time, reagents, and patient samples. Applications. Oncodiagnostics. Simultaneous detection of HER2, EGFR, Ki69 tumor-specific markers. Viral detection. Concurrent identification of CARS-CoV-2, influenza A and B. Immunological panels. Profiling of IgM, IgG, and cytokines in one assay. Benefits. Reduced sample volume requirements, faster turnaround time for comprehensive results, cost efficiency through reagent and resource optimization. Results. Cost efficiency, though reagent and resource optimization. DNA or RNA detection. Nanomaterials and nucleic acid analysis. Nanomaterials have revolutionized detection of nucleic acid sequences with unprecedented sensitivity and specificity. Hybridization probes. Nanoparticles, gold, silver, magnetic, are functionalized with complementary oligonucleotides that bind specifically to target DNA or RNA sequences. This provides molecular recognition with high specificity. Signal amplification. Probe-target interaction induce changes in optical or electrochemical signals, which are further enhanced by the nanomaterials. This dramatically improves detection sensitivity down to attomolar concentrations. Non-PCR methods. Nanoparticle-based sensors can detect nucleic acid sequences without the need for amplification, offering ultra-high sensitivity and isothermal detection capabilities for resource-limited settings. Point-of-care diagnostics. What is POC diagnostics? Diagnostic tests performed near the patient outside traditional laboratory settings, providing rapid results for immediate clinical decisions. Role of nanomaterials in field diagnostics. Nanomaterials enable miniaturization, increased sensitivity, and simplified operation for portable diagnostic platforms that can be used in resource-limited settings. Key components: microfluidic devices fabricated from paper, polymers, or flexible substrates modified with gold nanoparticles for visual colorimetric readout, magnetic particles for rapid sample purification and isolation, graphene or CNTs used as electrodes in electrochemical detection. Biochips. What are biochips? Biochips are miniaturized analytical platforms containing numerous probes, for example, antibodies, DNA, proteins, immobilized on a compact surface. They enable simultaneous detection of hundreds to thousands of targets in a single sample. Applications: genetic analysis, detection of SNPs, mutations, pharmacogenetic profiling, immunoassays, parallel evaluation of multiple antibodies or antigens, microRNA profiling, diagnostics for cancer and neurodegenerative diseases, forensic medicine, and legal investigations. Theranostics. Definition: Theranostics, from therapy plus diagnostics, is a concept that integrates diagnosis and treatment into a single nanoplatform. It represents a frontier in personalized medicine, allowing agents to both identify and treat pathological conditions. Components of theranostic systems: nanocarrier, liposome, nanogel, polymer capsule. Therapeutic agent, for example, chemotherapeutic, antimicrobial. Diagnostic marker, for example, fluorescent dye, MRI contrast agent, Raman probe. Targeting molecule, for example, antibody, aptamer. Clinical impact. Theranostic platforms enable real-time monitoring of drug delivery, therapeutic response, and disease progression, all while delivering targeted treatment, significantly advancing precision medicine approaches. Advantages of nanodiagnostics. Key advantages of using nanotechnology in diagnostics revolutionize healthcare by enabling more effective, accessible, and patient-centered diagnostic solutions. High sensitivity. Detection of ultra-low concentration of biomarkers, signal amplification through unique nanomaterial properties, low limit of detection, enables early diagnosis and screening, facilitates monitoring of disease progression. Miniaturization. Creation of portable and wearable diagnostic devices, reduced sample volume requirements. Automation. Integration with digital systems for real-time analysis, streamlined workflows, and reduced human errors. Limitations. Despite significant progress, nanodiagnostics face several challenges. Nanomaterial stability. Many nanostructures, especially organic or hybrid, tend to aggregate, degrade, or lose activity in biological environments. Scale-up complexity. Producing high-quality functionalized nanomaterials at industrial scale is technically demanding and costly. Need for standardization. The lack of unified protocols for evaluating efficacy, biocompatibility, and safety hampers clinical translation. Regulatory barriers. Nanodiagnostic products often fall outside current medical classification systems, complicating licensing and certification. Toxicity and safety. Potential risks. Accumulation in organs, for example, liver, kidneys, brain. Induction of oxidative stress, inflammation, or apoptosis. Unpredictable interactions with cellular components. Safety testing. In vitro. Cytotoxicity assays, ROS generation apoptosis analysis. In vivo. Toxicokinetics, histopathology, immune response evaluation. Alternative models. Organ-on-chip systems, 3D tissue cultures. Regulatory aspects. Compliance with OECD standard and ISO or IEC guidelines for nanomaterials in medicine. A growing need for legal updates at the level of WHO, EMA, and FDA. Ethical considerations. The application of nanodiagnostics necessitates adherence to key ethical principles. Informed consent. Patient must be fully informed about the technology risks and limitations, especially in cases involving implants or wearable monitoring devices. Data protection. As nanosensors are often linked to digital platforms, there is an elevated risk of exposure of sensitive health information. Bioethics. Technologies that involved genomic intervention, for example, CRISPR-based nanocarriers, must comply with principles of human dignity, transparency, and social justice. Technological equity. Access to high-tech diagnostics should not be limited to high-income countries or affluent populations. Role in low-income countries. Nanodiagnostics have the potential to transform healthcare access in resource-limited settings, overcoming infrastructure limitations and addressing public health priorities in developing regions. Cost-effectiveness. Mass production of paper-based nanoplatforms enables tests costing less than $1. Reduced need for expensive laboratory equipment and reagents. Lower operational costs for healthcare systems with limited budgets. Ease of use. Intuitive, user-friendly platforms requiring minimal training, no need for specialized laboratory infrastructure, accessible to healthcare workers with various levels of expertise. Mobility. Ideal for mobile clinics and field hospitals in remote areas, suitable for community health workers in rural settings, enables point-of-care testing in regions with limited transportation. Artificial intelligence and nanodiagnostics. Image analysis. Processing of microscopic or spectroscopic data from nanosensors. Detection of subtle variations and fluorescence signals or morphological features beyond human visual capabilities. Signal classification. Recognition of complex patterns across different biosensor outputs, differential diagnosis based on biomarker profiles, for example, tumor classification, pathogen identification. Predictive analytics. AI models use historical data to predict disease risks, treatment, response, or clinical outcomes from nanobiosensors readings. Automation. AI-integrated lab-on-chip platforms enable autonomous sample analysis and real-time result transmission to clinicians, reducing human error and accelerating diagnostics. Leading developments. Several pioneering institutions are at the forefront of nanodiagnostics research, developing innovative technologies that transform medical diagnostics and healthcare delivery. MIT, Massachusetts Institute of Technology, USA. Development of AI-enhanced nanoparticle-based diagnostics, smart nanoprobes for real-time disease monitoring. Harvard University. Paper-based CRISPR diagnostics for point-of-care applications. Ultra-low-cost diagnostic platforms for global health. Oxford University. Graphene-based biosensors for continuous monitoring. Nanopore technology for rapid DNA sequences and analysis. National Academy of Sciences of Ukraine. Advancements in magnetic nanoparticles for diagnostics, localized diagnostic platforms for infectious diseases. Clinical trials. Target patient groups. Cancer patients, evaluation of sensors for PSA, HER2, CA125, detection of tumor-derived microRNAs in blood, saliva, and urine. Diabetic patients, continuous glucose monitoring using flexible graphene sensors. Subcutaneous implantable nanogels containing glucose-specific enzymes. Infectious diseases. Gold nanoparticle-based test for COVID-19, tuberculosis, syphilis. CRISPR-enabled nanoparticle platforms for rapid specific viral diagnostics. Patenting and technology transfer. Patenting. Protection of intellectual property for biosensors, nanomaterials, and functionalization methods. Patents secure commercial rights and incentivize research advancement in nanodiagnostics. Technology transfer. Licensing of nanomaterials, for example, AuNPs, functionalized with aptamers, to medical device companies. Industry partnership, Roche, Abbott, Thermo Fisher actively acquire nanomedicine startups to integrate novel diagnostic technologies. Funding mechanisms. Crowdfunding platforms, for example, Kickstarter, Indiegogo for personal diagnostic devices. Venture capital investment from deep tech and digital health VC funds drives innovation in nanodiagnostic commercialization. Key concepts, essential terminology and concepts in nanodiagnostic technologies. Nanomaterials, engineered materials at the nanoscale 1 to 100 nanometers with unique physico-chemical properties. Biosensors, devices that convert specific biological interactions into measurable physical signals. Quantum dots, fluorescent semiconductor nanoparticles with size-dependent optical properties. Liposomes, lipid-based vesicular structures used for drug delivery and biomarker detection. Microfluidics, systems that manipulate fluid at micro to nanoliter scale through engineered channels. CRISPR diagnostics, gene editing tools adapted for ultra-sensitive molecular detection of specific nucleic acid sequences, make them superior to conventional dyes. How are they used in multiplexed analysis? What are the bioethical risks associated with nanomaterials in medicine? How can microfluidic platforms transform healthcare in developing countries? What research directions are most promising for Ukraine in nanodiagnostics? How can AI assist clinicians in decision-making based on sensor data? Recommended resources. Scientific reviews. JNKK, Nanodiagnostic Application of Nanotechnology in Molecular Diagnostics. WENC, Electrochemical Biosensor, Recent Advantages and Application Chemical Reviews. Open access articles. NanoBioSensor, Current Advances and Future Perspectives in Diagnostics, Biosensors and Bioelectronics. 2021. CRISPR-based diagnostics, Emerging Applications in Infectious Disease and Oncology, Nature Reviews, Genetics, 2022. Online resources, PubMed Central, Free Database of Biomedical Literature, Clinical Trials Go, Registry of Clinical Trials in Nanodiagnostics. Recommended sources, Scientific Literature, Key Books and Reviews, JNKK, 2020, Nanodiagnostic Application of Nanotechnology in Molecular Diagnostics. Wang J, 2019, Electrochemical Biosensor, Recent Advances and Applications, Chemical Reviews. Open access articles, Nanobiosensors, Current Advances and Future Perspectives in Diagnostic, Biosensors and Bioelectronics, 2021. Quantum dots for multiplexed biosensing, a review, analytical and bioanalytical chemistry, 2020. CRISPR-based diagnostics, emerging applications in infectious disease and oncology, Nature Reviews Genetics, 2022. Technical resources, patents. US 104506889B2. Gold nanoparticle-based lateral flow immunoassay. EP3709901A1. Graphene-based biosensor with electrical readout. Online resources. PubMed Central, free database of biomedical literature. Google Patents, search engine for global registered technologies. Clinical Trials Go, Registry of Clinical Trials in Nanodiagnostics. Cordis, EU, research platform for Horizon Europe projects.